Welcome back to this second day of our series of lectures on the genetics of the immune system. And last time we looked at the basic components of the immune system and especially those specific cells called B cells that morph or differentiate into plasma cells. And then those plasma cells are capable of producing these little Y-shaped protein molecules called antibodies. And then we talked about how antibodies are sort of like little missiles that are shot out or produced by and shot out by the plasma cells. They're spewed out by plasma cells, and these little antibodies act like sort of like little missiles then that seek out and attach to the surfaces of invading viruses or bacteria, which can then mark those viruses or bacteria for destruction by different components of the immune system. And what I want to look at right now with you is I want us to take a look in detail at just what an antibody is, what it's made of, and how they operate. So we've got a diagram here on the slide of the structure of an antibody, and if you'll look on the top of page 5 of your study guide, you'll see a very similar diagram with blanks so that you can go ahead and label these parts yourself. So I think right now, why don't you go ahead, pause this video, and label your diagram at the top of page 5 so that it looks somewhat similar to this one on the screen. And I'll give you a little bit of time to do that. And so go ahead and pause the video, and then we'll come back and we'll continue on with the story. Okay, so let's dissect apart this antibody molecule. Remember I said it's a little Y-shaped protein. This is the way most antibodies look. There are variations on these, but this is kind of the basic kind of antibody. And as you can see in this picture, or you'll be able to see as we go along, this antibody is really made up of 4 different protein molecules joined together to produce this bigger Y-shaped protein called the antibody. And let's take a look at the parts one by one. For example, here you can see the black arrow on the screen is pointing to what's called a light chain. And the reason it's a light chain is because it's a relatively short chain of amino acids. So remember, that light chain, that's a protein. It's a string of amino acids. And if we move ahead here, you'll see that this particular Y-shaped antibody molecule is made up of 2 light chains, and also 2 what are called heavy chains. And if you'll look at the heavy chains, you can see, I think, that a heavy chain is actually made up of, if we go back here, let's go back to this. Okay, that heavy chain right there with that most recent arrow that appeared is made up of a chain that is about the same size, you can see, as a light chain, but then it looks like it's hooked on by looks like a little springy wire or something. Those are actually disulfide bonds holding that upper chain of the heavy chain to the bottom portion that's going straight up and down vertically. And so together they make up what's called a heavy chain. And as you can see, just like we have 2 light chains making up this antibody, we also have 2 heavy chains. So the antibody is really made up of 4 different protein molecules joined together. 2 light chains, 2 heavy chains. Now, I want to point out something here about this arrow on this screen, pointing to what's called the constant region of the light chain. That's a sequence of amino acids that is the same in all different kinds of antibodies. it's the same, and that's why it's called the constant region. And if you look at the upper portion of that light chain, you'll see there's a portion called the variable region of the light chain, and that is a sequence of amino acids that can vary, that does vary from one kind of antibody to another. And so I think you can see here that there's an upper region of the heavy chains also that's composed of a variable region. And so if you'll look at the variable region of the light chain right next to the variable region of the heavy chain, you can see that that portion, the two prongs, if you might think of it that way, as of that Y-shaped protein molecule, the two prongs, the antigen binding sites is what they're called. Those are variable regions. In other words, this antibody has a three-dimensional shape. That antigen binding site, therefore, will have a particular three-dimensional shape that will specifically fit a particular antigen molecule on the surface of a particular invading virus or bacterium. So the variable regions are going to be just that. They vary in their three-dimensional shape. So, for example, a couple weeks ago I got over a really bad cold. And since I'm over it now, what that means is in my body I have antibodies that were produced during that infection that have a specific three-dimensional shape of their antigen binding sites that perfectly fit the antigens on the surfaces of the viruses that I was infected with a few weeks ago. They fit them perfectly, like a lock and key, like a hand in a glove, so that if I'm ever invaded or infected by that particular kind of cold virus again, I will be immune to it. I won't catch it because I will already have all of these memory B cells, remember memory B cells, that have the ability to morph into plasma cells to produce those specific kinds of antibodies with that specific type of antigen binding site that fits perfectly those antigens on those viruses that I was infected with a couple weeks ago. Does that make sense? Boy, I hope that makes sense. So each kind of antibody is very specific, and it will only fit one particular type of antigen on one particular kind of pathogen, whether it's a virus or a bacterium. They're very specific, very specific. We have the potential, the ability within our bodies to produce hundreds of trillions of very specific kinds of antibodies, each kind of antibody capable of latching onto a particular antigen on a particular germ. So right now there's a lot of talk that you might be hearing about hopefully that we can begin to develop antibody tests for COVID-19, the novel coronavirus. If somebody's infected by coronavirus and they recover, they're still going to have antibodies in their body that were produced during that infection that specifically bind to particular antigens on the surface of the COVID-19 virus, making them now immune to COVID-19. And the reason scientists are excited about coming up with this antibody test is because it's a quick and easy blood test, quick and easy, and they would be able to do millions of people, test millions of people throughout the country to find out who's had it maybe and didn't even have symptoms, and then you see we can get a better idea of what the actual death rate of the virus is. Because if we find out that, let's say, 10 times more people have been infected with the virus than what the virus tests actually show us, that would drive down the death rate. So it's important for us to find out that information. Okay, so these are the main parts of an antibody. And again, I want to point out that the heavy chain, the reason it's called a heavy chain, made up of two parts, you see, joined together by kind of a springy-like looking thing. The reason it's called the heavy chain is because it's like twice as long as a light chain, and so it's a heavier molecule. So that's kind of the anatomy of of an antibody. Now, let's look at how antibodies form. I'm looking at the middle of page 5 in your study guide, and I want to particularly focus on letter C there in the middle of page 5. The big question: how can only 200 different antibody genes that are responsible for making antibodies, there's only like 200 of them, how can they produce 100 trillion different kinds of antibodies? I mean, my goodness, we have the potential within our bodies to make like 100 trillion different specific types of antibodies, but we've only got 20,000 genes. Maybe only 200 genes responsible for controlling the production of antibodies. So how are we able to generate all this antibody diversity no matter what kind of disease-causing organism might someday invade our body? Well, it's pretty cool how this works, pretty ingenious. And it kind of reminds me a little bit of mixing and matching outfits. Some people have a lot of different outfits in their closet, and some people don't. I have, several different kind of pairs of pants that I'll wear to school when we're together in school. Jeans on Friday, yay! But then maybe there's some khaki slacks hanging in the closet, maybe some blue slacks hanging in there, maybe some brown ones, and all these different colors of shirts. And so maybe one day I might wear khaki pants with a red shirt. Maybe another day I might mix and match and wear those khaki pants with a blue shirt or something like that, kind of mix and match. This is what the generation of antibody diversity reminds me of. Now, on this particular screen of the presentation, and I'm sorry in your study guide it's not in color, but you can kind of follow along here. But if you'll look here, we're looking at one of maybe 200 different kinds of antibody producing genes. And I'm only talking about the ones that produce the heavy chain. And keep in mind there's a whole bunch of other genes in addition to these 200 or so that are responsible for just making the light chains of the antibodies. But let's look at the parts of a gene for the heavy chain of an antibody. So I'm looking at this sequence of DNA right here that the black arrow is pointing to. And notice that this gene is made up of a whole bunch of different segments, different parts. There are 3 different V segments or V regions, and those that stands for variable regions. There are 2 different kinds of D regions in this gene, that stands for diversity. And there are 4 different kinds of J regions, and J in this case stands for joining region. And then C is for constant. C is the portion, the blue C region of that gene is the portion of the gene responsible for making the constant region of the heavy chain of an antibody. What happens is, let's say we're looking at this particular gene inside of a B cell, a newly formed B cell that will eventually develop or differentiate into a plasma cell that will produce these antibodies. So we're looking inside of a particular B cell. This B cell has just been born. It's just come out of the bone marrow where it was produced. And now something really interesting happens. Mixing and matching. Shuffling of the gene components. So what happens is specific enzymes actually take that gene represented by the black arrow. Specific enzymes take that gene and randomly snip out one of those 3 V regions, and then they randomly snip out one of those 2 D regions, and it's random, and then randomly snips out one of those 4 J regions, and then snips out that C region. There's only one C region, that constant region of that gene. And then other enzymes that act kind of like glue those pieces of that gene together to produce an entirely new gene represented by this black arrow right here. The rearranged gene components. A newly manufactured, it's like a natural recombinant DNA going on here. A new gene that gene will then code for the heavy chain of the Y-shaped antibody molecule on the right-hand side of the screen. And so from one B cell to the next, the components that make up this rearranged gene shown by the black arrow, from one B cell to the next B cell, they're going to be different. And what makes them even more different, creates even more possibilities, is, remember, this is only one gene we're looking at here, only one heavy chain gene out of 200 or so. And so you get all this shuffling going on. And then if you layer on top of this all the genes, which we're not even looking at here, that produce the light chains. We just looked right here at the production of a heavy chain. If you add on to these 200 or so genes, all the hundreds of genes that produce the light chains, you can imagine how we're able then to produce almost an endless supply, endless variety of different unique antibodies. And you can kind of see that if you think about it. I mean, every cold that you've ever caught in your life was caused by a different, unique cold virus that your body had never been exposed to before. So the last cold you caught, you will not catch that particular cold virus again, because you now have the antibodies that were specifically made for that particular cold virus when it infected you. So the older you get, the more immune you're going to become to lots and lots of different things. So this is kind of how all these different varieties of antibodies are produced. Now, I was thinking about, what this reminds me of, the production of endless varieties of unique kinds of antibodies, this mixing and matching. And I thought, gosh, that reminds me of something. What does that remind me of? Now you got to bear with me. I've got grandkids. So my mind started thinking, oh yeah, I know what this reminds me of. It reminds me of Mr. Potato Head. So think about this, okay? Divert your attention away from actual antibodies for a minute to this model here, which can kind of help us understand how we can create lots of different kinds of antibodies. Imagine with Mr. Potato Head with 5 different kinds of eyes, and let's see, 3 different kinds of feet, and 3 different kinds of hats, and what are those, noses? 4 different kinds of noses. 1, 2, 3, 4, 5, 6, 7, 8 different kinds of mouths, 2 different kinds of ears, and 1 common region right here. With all of those plus this 1 common region, I wonder how many different kinds of antibodies we could make. That is Mr. Potato Heads. Well, I started playing around with this, and I thought, well, we could make one that looks like this, or like this, or like this, or like this. Oh my goodness, the possibilities are endless. And see, no two Mr. Potato Heads that I'm showing you are identical. Each one is unique, just like each kind of antibody is unique. That's how it works. That's how it works. Ooh, how much? Haha, okay, that was the last one. Yeah, I think you get the picture. I think you can get how this works. So now, let's look at where we've been. There are these two main branches of the immune response. And up until now, we've already looked at what's called the humoral response, the right-hand side of this particular diagram here, the humoral response. And that's the part that involves macrophages. Remember, those are the first responders, the macrophages. They're the first ones that start attacking the enemy. The macrophages talk to the B cells, which are trained. Well, I'm sorry, let's back up. The macrophages talk to the T cells, which then train B cells. And then the B cells, some of those B cells hang back. They become memory B cells. And other B cells will then differentiate into plasma cells that produce specific kinds of antibodies. And you can see that on the right-hand side of the diagram. Something that's real important to talk about that we really haven't emphasized yet is if you'll look at where the red arrow is pointing to that B cell, that B cell has been selected among millions of different kinds of B cells because it produces, just by chance, a particular shaped antibody that will match perfectly the antigens on the surfaces of the germs that the macrophage has identified. And so that particular B cell will start doing mitosis like crazy, producing lots and lots more identical B cells that will all make the exact same kind of antibody that will attack that particular specific germ. This is sometimes called B cell cloning. It's undergoing rapid mitosis. And of course, you can think of this as kind of increasing the troops, the number of troops. So the number of B cells are increasing dramatically in the body here, and that's part of the reason why the lymph nodes might start to swell. Okay, so that's the right-hand side of the diagram, the humoral response. What we've not looked at yet is the left-hand side of the diagram, called the cellular response, and that's the next topic in the study guide. So if you'll turn over to page 6, top of page 6, we're going to begin with this other branch called the cellular immune response. And if you'll notice on the top of page 6 in your study guide, it starts with macrophages again. The macrophages are ever-present, reliable, kind of the first line of defense, you might say, that they eat the invaders and display the invaders' antigens on their surfaces, kind of like trophies. And then the macrophages present these trophies to the T cells, specifically helper T cells. And on the left-hand side of the diagram on the screen, you can see there's a T cell right there. Right there he is. And the T cells are going to do two things here. They're going to follow along where it says cytokines. Those are chemicals produced by the T cells that actually make contact with B cells, communicating to the B cells, okay? This is how the T cells train or program the B cells. But at the same time, and I want to focus on the left-hand side of this diagram now, at the same time, those helper T cells are going to talk to different kinds of T cells called cytotoxic T cells, and you can see that in the diagram there. Cytotoxic T cells, sometimes they're called killer T cells. And they kind of remind me of, I don't know, Navy SEALs, because they're amazing. They can kill in a variety of ways. They're highly trained. They can kill germs or cancer cells, which are just normal cells that have gone rogue in lots of different ways. They're multi-talented, these cytotoxic T cells. They're not specific like B cells are. Remember, B cells only make one specific kind of antibody. Each B cell makes one specific kind of antibody. Cytotoxic T cells, on the other hand, can kill in a variety of ways. So if you'll look at the top of page 6 in your study guide, go ahead and write down the definition there for macrophages. And we talked about them before, so I think you probably remember those guys. They're like the first defenders that attack and actually literally eat the invading viruses or bacteria and then display the antigens on their surfaces, a little bit like a trophy. They present those antigens to helper T cells to alert the helper T cells that there's a battle brewing and the troops need to be roused. Number 2 on page 6, T cells. T cells on the left-hand side of the diagram, the cellular response. You can write down for number 2 that T cells differentiate or develop into killer T cells. Actually, more specifically, there are different kinds of T cells. Let's back up just a little bit. The helper T cells talk to the cytotoxic T cells, which then become killer T cells. And then number 3, cytotoxic or killer T cells, they will then in turn kill whatever has been invading the body, whether they're viruses or bacteria, or like I said, cancer cells that are just basically normal cells that have gone haywire or have gone rogue. And there's a killer T cell right there. Now, you've got this diagram on page 7 of your study guide, and this just illustrates a little more clearly what we just looked at. If you'll look at this diagram, and it's just like the one on page 7 of your study guide, right here where this first black arrow is, we've got a macrophage engulfing or eating this sort of an orange-colored pathogen. Maybe it's a virus. An orange-colored pathogen that's got these little blue dots on the surface. I think those are blue, and those are the specific antigens on the virus. And the immune system here is recognizing that those antigens are foreign. And so the macrophage envelopes and digests and literally eats this virus. And then if you'll move on down the diagram, you can see in the next picture just to the right of that first macrophage, the virus being digested inside the macrophage and a virus antigen being displayed on the surface of the macrophage. And then moving on down in the diagram, you can see the macrophage connecting to, talking to a helper T cell and presenting that antigen that was on the virus, presenting it to the helper T cell, basically saying, look at this trophy I've got. Look at this antigen that I've got from this virus that I just ate. That gets the T cell, the helper T cell, all excited. The helper T cell then produces little molecules that actually attach to, over here, virgin, that means brand new, or memory cytotoxic T cells. Virgin or memory killer T cells. And that tells them which particular invader they're supposed to go after. And if you look further down the diagram, you can see the cytotoxic T cells, then they start going through mitosis. And that's very important because we're making millions and millions more of those particular kinds of cytotoxic T cells. They differentiate and become what are called effector cytotoxic T cells, or you might say sometimes they're called killer T cells. And then look what the killer T cell is going to do in turn. It's going to reach out, grab onto this infected body cell. Now, this is a body cell. It's a normal body cell that belongs to this person, but it's a goner. It's all swollen because there are millions of viruses inside of it. It may have theoretically originally been invaded by one virus that got inside that body cell, and that virus brainwashed that body cell's DNA in turning that body cell into a factory that has manufactured millions of new viruses, and they're about to blast their way out of this swollen body cell. And each of those new viruses that emerges from that normal body cell will go and infect another body cell, and the infection will take off exponentially. So what's going to happen here is, since that body cell is all swollen and full of viruses that are about to emerge, notice that the killer T cell, that blue killer T cell, will kill that body cell before those viruses have a chance to emerge from the body cell and go and invade other body cells. And so right here you can see then that body cell has been destroyed by the killer T cell. And that's what happens. Now here's a close-up view of a cytotoxic T cell, in other words, a killer T cell, in hand-to-hand combat with a cancer cell, you might say. So we've got on the left-hand side of the diagram, we've got a cancer cell there shown in red or pink, whatever that is. And it's got these foreign antigens on it. The foreign antigen, notice, is the little orange structures on the surface of that cancer cell. So if we go back in time, that cancer cell used to be a normal human cell, a normal body cell, but there was some kind of a mutation in the DNA within that cancer cell, causing that cancer cell to go haywire, to go rogue. And when cancer cells do that, when cancer cells develop in the body, they produce new kinds of antigens on their surfaces that all of a sudden are foreign to the body's immune system. And so this cytotoxic T cell has recognized those antigens on the surface of that cancer cell. They may only be a few atoms different from the normal body cell's antigens, but that's enough for the immune system to recognize, hey, these are foreign antigens on this cell, which we call a cancer cell. I need to attack this thing. So in this diagram, you can see this cytotoxic or killer T cell in hand-to-hand combat with this cancer cell. In the middle picture, moving over to the right, in this example, in this particular example, this cytotoxic T cell is releasing these protein molecules called perforin, and it seems like a very appropriate name because those protein molecules actually perforate or punch holes in the cell membranes of that cancer cell. And in the third picture to the right, to the far right, you can see that cancer cell is lysing, it's dying, because the cell membrane's been all punctured by these perforin molecules. And that just shows one way that killer T cells are able to kill and protect us. Here's a photograph taken through the electron microscope of a cancer cell in yellow or orange being attacked by a whole bunch of things that look like little white puffballs, but those are not puffballs. Those are actually killer T cells, all ganging up on this cancer cell. And kind of reminds me of, I don't know, maybe a pack of wolves ganging up on a moose or something like that. but you can almost see the action in this photograph, even though it's a still photograph. So there we have it. We are ready now for the top of page 8 in the study guide. But I think that for today, this is a good place to stop. And so we'll begin with the top of page 8 in your study guide next time. So have a good day.